Yes,
close
Koyuncu.
enough, to our CVR seminar series. Orkide arrived in January this year, and we are so thrilled to have her. She's a fellow herpes virologist, and not only just a fellow herpes virologist, but a fellow alpha herpes virologist. So Orkide's work is really focused on understanding changes in, I'm not going to butcher your field of study, but neural biology, and using viruses both as tools to study that, but also as tools for disturbing that homeostasis. And so she's very interested in both host pathogen interactions as well as just broadly speaking neurobiology and neuropathogenesis. She got the amazing opportunity to train with Dr. Lynn Inquist, who many of us know and is a wonderful and generous colleague. Who has supported many careers, including several on this call. And she comes to us with a nice set of publications already in R21, and hopefully she's hitting the ground running and not getting too many obstacles due to COVID. I'm really sad that we could not do this in person, but I think there is a light at the end of the tunnel. And so we welcome you. We really are thrilled to hear more about your work. And then we're all going to stay on after the talk and just discuss other things that we could do to help you get things launched and just have informal discussion. So I won't take any more of your time, Orkide, and I will hand the center stage to you.
Thank you so much, Ilham. This is a very nice welcome, and I will share my screen. And please let me know if everybody's able to see it. Okay, so I am very happy to be here today and present from UCI as a part of the MMT department, almost a year after my first interview. And oh, what a year it was. So today I will talk about nervous system infections and basically about viruses taking the long way. But I wanted to have a broader start looking at also other viruses in the nervous system. Virus infections usually start in the peripheral tissues, and many of them are mostly retained or cleared in these peripheral tissues. And most of these infections do not even enter the nervous system, not only because of the multi-layer barriers protecting the nervous system, but also it doesn't seem to be a good strategy to replicate in neurons, because most of the time this leads to the death of the host, which actually also represent a dead end for the pathogen. But many of the viruses, they can find their way into the nervous system. Most of the time they are newly emerging zoonotic infections, and they do so most of the time by permeating the blood-brain barrier. Usually the endothelial cells, the brain microvascular endothelial cells, they have these tight junctions. They do not let pathogen or micromolecule transfer. But sometimes viruses like HIV, SIV, they can infect the blood monocytes, and they can pass this barrier by hiding in these cells. Sometimes some viruses, they can infect these endothelial cells, and this is how they find their way into the central nervous system. Sometimes the respiratory tract infections, these viruses can infect the olfactory nerve from the nasal cavity, which is basically one synapse away from the central nervous system. In laboratory conditions, many of the viruses can infect through this route, but even the recently emerging SARS-CoV-2, there are some indications that it can enter the nervous system through this route, or maybe through the blood-brain barrier. But these are most of the time, as I said, the accidental pathogens. But there are two virus families which actually infect the nervous system as a strategy, as a life cycle strategy. And they do so by exploiting the biology of these neurons. Alpha herpes viruses that I'm going to talk mostly about today, they infect the sensory neurons of the peripheral nervous system, and they can invade these axons, and they are trafficking in a retrograde manner. they establish an infection in the ganglia, and after reactivations, I will talk about in detail later, they can also traffic in an anterograde manner. This is unique mostly to this viruses, alpha herpes viruses. Rabies virus can invade the motor neurons through neuromuscular junctions, and the particles can go in retrograde manner. And they follow the synaptic connections, and they can reach the central nervous system that way. So this is why I'm very interested in studying these viruses that I say taking the long way, because they have to traffic in these long axons, especially alpha herpes viruses when they infect the peripheral nervous system. So I believe I don't need to give a deep introduction into herpes viruses to this audience, but I want to remind you certain key facts about these viruses. We know that they adapted their host very well. They have more than 100 million years of coevolution with their host. Not only humans, many mammals have their own herpes viruses. Well, except mouse and rat, they do not have their alpha herpes viruses. This makes our life a little more complicated. But so humans have herpes simplex virus 1, 2, and VZV. And these viruses are double-stranded DNA viruses. They have an envelope, and they have a characteristic tegument layer in between the envelope and the nucleocapsid. They are fairly large, around 200 nanometers. And their winning strategy is not that they can infect many of the cell types productively, but they target the nervous system to establish a lifelong reactivatable persistent infection, what we call latency. This strategy makes them one of the most prevalent pathogens in the world. I will give you an example about HSV1. So according to a report that was published in 2015, there is approximately almost 4 billion people under the age of 50 that are HSV positive. And we know that this number is much higher than that. And we expect it to be around 70–80% positivity in population. And usually if the immune system is strong, these infections are very well controlled. And the initial infection and the reactivations, they represent as mild oral lesions, as we call them, cold sores. Less frequently, if the eye epithelium is infected, it can lead to herpes stromal keratitis, which can lead to blindness. And very rarely, if there are certain mutations in the immune system, or if the patients are immune compromised, virus infection can spread to the central nervous system, which is indeed a very nasty disease, and it is called herpes simplex encephalitis. But the life cycle is indeed remarkable. It starts, I will give again the example of HSV1, and infection starts in the epithelial cells of the oral mucosa. And there, each infected epithelial cell can produce hundreds and thousands of progeny in a very short amount of time, and some of them can invade the axons of the sensory neurons of the trigeminal ganglion. There, the virus particles can traffic retrogradely to the cell body, and when they reach the neuronal nucleus, now they start a different type of infection, and this time they do not produce infectious virus particles. The viral genomes are silenced, so they are circularized, they are covered with silencing histone modifications, and they stay lifelong in these neurons. There is no cure right now to get rid of these episomes from the infected neurons. By the way, there is also no protective vaccine against HSV1 and HSV2. So this is how, when they infect, they stay forever. And upon reactivations, they can produce infectious virus particle that can travel back to where they entered the organism. And there, as soon as these epithelial cells get infected by a few virus particles, they can amplify it immediately. And this is how they find their way to the new host. So they are never leaving the host, and several times during latency, they can find their way to new hosts. Okay, so many of the early events, starting with the spread of virus to the nervous system, then after reactivation, the spread of other hosts, we do not know some of the main mechanisms because it is not easy to study their interaction with the nervous system. One of the reasons for that is that the end of axons of peripheral nervous system is centimeters to almost a meter away from the PNS ganglion where the neuronal nuclei are. And these axons, not only in contact with these epithelial cells, but they are also sensing the infected cell milieu when these epithelial cells are in fact productively infected with these virus particles. So when I started working with these viruses, with neurons, I wanted to understand how do these axon termini respond to viral infection, inflammation in the peripheral tissues. And I was very interested in understanding whether this response have an effect on how the infection is established in the neuronal nucleus. So here again, I would like to stress one more time that the entry and fast retrograde trafficking in axons is the key to establish a successful infection in neuronal cell bodies. And here again, I use the example of these two viral families. One is alpha herpes viruses, the other one is rhabdovirus. Here I try to illustrate a rabies virus. They enter the nervous system in a very different manner. Alpha herpes viruses, they use receptor-mediated membrane fusion, and upon the fusion of their viral membrane with the axonal membrane, nucleocapsids are released by themselves in the exoplasm. And several thousands of copies of the tegument proteins are also released. So they traffic by themselves. They have to engage the dynein motor by themselves. On the other hand, rabies virus particles, they're endocytosed. So they are being transported on the axonal microtubules by dynein, but they are still in the endosome. Basically, they are still outside. This is a key difference between these virus families, and I will use this difference in the following slides. So to be able to study this directional infection, my model system uses the superior cervical ganglionic neurons. In humans, here is the superior cervical ganglion. This is the TG, so it's below the TG. And in mice and rats, you can find the SCG tightly wrapped with the nodose ganglion, and you can see the valgus nerve in between. So these neurons, they have robust axons. As you can see with the actin staining in this picture, they have a large globular cell body, and their nucleus is on the other side of the axon. Here you can see it in green, and their TG is just all around the cell body. You can see it as a dispersed staining. So when I see these neurons as a dissociated culture, they grow robust axons, and sometimes they bifurcate. In vitro, they do not grow dendrites. So this is to my advantage because all the neurons that I can see in the dish are basically axons. So you can see the cell bodies and axons filled with GFP protein. But as it is not very ideal for me to study directional infection, because when you infect the neurons in dish like that, the infection goes to productive, and the virus particles can enter either from axons or from the cell bodies. To be able to study the axonal spread, I model this axonal infection by using a modified Campanella chamber system that includes 3 compartments. Basically, I took the embryonic SCG neurons, and I see them in the soma compartment that is depicted as S compartment here. And they form these nice clusters that can grow robust axons. But the trick is to guide them. If you don't guide them, they make circles around. But if you etch grooves on the plastic surface, they follow these grooves, and they can grow axons resembling to a nerve fiber, and they can pass two silicon barriers. And at the end of 2 weeks of maturation, you can have really nice axonal compartments. Most of the time I call them N compartment from neurite compartment, and the middle compartment is called the M compartment. So this is an actual picture of such a chamber. And this is very ideal because it enables isolation of neuronal cell bodies from axon termini, and it enables me to do directional infections, and I can see the spread either from axon ends to the cell bodies or from cell bodies to the axons. And the other advantage is that we can do nice live cell imaging on these compartments, and biochemical analysis of axons. We can check new protein synthesis, we can do mass spec, RNA-seq experiments. And in many of my experiments, I used pseudorabies virus. This is a pig pathogen, but it has a wide host range. It infects most of the mammals except higher primates. It is very similar to HSV1. It is neuroinvasive. It can be used as a circuit tracer. So during my talk, you will hear me talking about axonal infections most of the time. So I wanted to remind you that axons of the peripheral nervous system can take up to 99% of a neuron cytoplasm. So they are pretty large and long compared to the CNS axons. So this is why efficient long-distance communication is essential for their homeostasis. And this is why axons respond to various external stimuli by local protein synthesis. So local protein synthesis now is well accepted and known, especially in the central nervous system. We know that it is required for synaptic plasticity and memory storage, but for a long time, it was a very hotly debated area of research, especially in the peripheral nervous system. But now we know that it is required for axon guidance during development and responsiveness during regeneration, and also during the communication between cell body and the distal ends, especially during injury, where retrograde signaling is essential for repair and continuation of the life of that axon. So I was very interested in that phenomenon when I started, and I asked the question, what happens when herpes viruses invade the axons? And yes, the answer is that if we inhibit protein synthesis only in axons, virus infection is indeed not very efficient. So I will use this fluorescent infectious unit assay throughout my talk. So I would like to introduce it to you. I make my infections in the axonal compartment most of the time, and either before or after infection, you can add different translation inhibitors or drugs to this compartment. And most of the time, I also like to see how many of my neurons are connected to the end compartment, because not all of them can send axons passing these two barriers. So most of the time, you will hear me using a fluorescent dye. If I'm using a red capsid virus, I'm using a green fluorescent dye to label the connected cell bodies. So each of these dots in the screen you see are neuronal cell bodies, and if they are red, it means that they're infected. So this is a very basic system that I can test many of the things. And the first things, as I said, that I tested was a translation protein synthesis inhibitor. In this case, you see cyclohexamide. And even when only axons are treated with the protein synthesis inhibitor, in the cell bodies, I could see that the infection rate was reducing a lot. And if cell bodies are treated with this inhibitor, of course, it immediately blocks infection, because the virus replication requires new protein synthesis. And I was able to show that the inhibition of protein synthesis in the axons actually leads to affects particle motility, and more particles are stalled in axons. They are not moving, basically. So it happens right after they enter and start moving. Then we asked the question, what proteins are made in axons after virus infection? We did click labeling and a mass spec approach at that time, and we found several proteins that are made. And I will not go into detail, but I would like to stress that there are certain proteins that we picked. 3 of them are peripherin, which is a type 3 intermediate filament. The other one is annexin A2, which regulates endocytosis and exocytosis. And the other one is a dynein regulator, LIS1. And all of them had effects when I did RNA knockdown in the axonal compartment, and they both reduced virus infection. But more interesting to me, and I would like to stress here that this pathway had a very high similarity to the retrograde injury signaling, because many of these proteins resemble to the pool that was made after the injury. And not only the identities of these proteins that are made, but what is happening right after injury and virus infection resembles to each other very much. For example, there's a calcium increase right after infection and injury, and both these retrograde injury signaling molecules and viral nucleocapsids, they need fast retrograde transport that is targeted to the neuronal nucleus. So I wanted to test the hypothesis that whether injury or virus infection can compete with each other if they are using the same pathway. So in this experiment, I will show you virus trafficking in healthy axons. This will be the A portion. And then I will show you how the virus trafficking is affected by a cut in the axons. I will show you both the proximal axon that is still connected to the cell body and the distal axon that is cut out from the cell body. If you look at the virus trafficking in uncut axons, here I'm using a green capsid virus. You can see trafficking in 10-15 minutes after applying the inoculum very nicely. And if you look at the proximal cut axon, you see something like that. And this resembles a lot when I use a protein synthesis inhibitor, cyclohexamide or emetin. So particles, still you can see some moving ones here and there, but they're mostly stalled and they're kept in the axon. And to my surprise, when I look at particle trafficking at the distal axon, I saw this. Even after 1 hour, particles still can traffic in the cutout piece, but not in the proximal piece. And here is a graphical showing that observation. So this is my chamber. Here is where I'm making the cut with a scalpel. And these are the moving particle percentage in control axons and distal axons. And I see a significant decrease in the proximal axon segment. And if you observe these axons, this is how they look, very nice, uncut axons. And 1 hour after the cut in the proximal segment, they start becoming less healthy. They look like they have blabbing and several varicosities. And if you look at the distal segment that is not connected to cell body, yes, the ends are sealed, but you can see the axons are still looking healthy. So there is something happening in the proximal segment that is also interfering with the virus trafficking. So this led me to a conclusion that if healthy axons are infected, there is local protein synthesis and there is efficient retrograde transport of virus particles. And when these axons are cut, there is retrograde injury signaling that's already happened, and now virus particles are stalling. And this means that pre-injury competes for efficient PRV retrograde infection, and this probably means that infection induces and may be repurposing the axonal damage response to reach the neuronal nucleus in a timely manner. So that was very interesting to me, and I started asking questions that in an animal, in the tissue, these axons are already exposed to the infected epithelial cell environment. And I was thinking maybe that could act like an injury signal for them. And I asked questions, what happens when these axons are treated with interferon, because these infected epithelial cells produce copious amounts of cytokines. So I wanted to test how these axons would respond to cytokines and how this response affect retrograde particle trafficking, and if there is a way to see how these responses are regulated. So when I started these experiments, there was a very talented grad student who started rotating with me. And she took over the interferon experiments. And we found that immediately after treating these axons with interferon, either with interferon beta or interferon gamma, there was a reduction in the retrograde infection of PRV. We did the same experiments as I showed you with the injury and the cyclohexamide, and we looked at particle transport in axons immediately after infection, and we saw that indeed in interferon beta and interferon gamma treated axons, particles are not moving efficiently, they are stalled. So each line represents moving particles because these are stacked images of movies. And if you see more dots, it means that they are stalling. There are sometimes little movements here and there, but it's not efficient. And if you look at the graphs, it's more or less reduced to half by half. So then I asked the question, is it the same in different viruses? And, always I want to compare it to the rabies virus, the other virus family that's taking the long way. And at the time, another very talented MD PhD started rotating with me, and we started asking these questions. And at the end, she even went to the Schnell lab and made an M-cherry coupled P protein expressing rabies virus so that we could track each particle moving on the axon. So we used this M-cherry rabies virus, and we followed the same experiments I showed you in the previous slides. And to our surprise, this time we saw that treating axons in the N compartment with interferon beta or interferon gamma had no effect on the retrograde virus infection efficiency, as you can see from the pictures here and from the graph here. Of course, if you treat the S compartment cell bodies with interferon, you significantly reduce virus infection. So that was very interesting, and that showed us that maybe there is something going on specific to the alpha herpes viruses that are entering the axons via membrane fusion. And then we asked whether the regular cargo transport is affected in these axons when we treat them with an interferon. And we did this experiment with Jen. We looked at lysotracker positive puncture transport, and this is particle trafficking or lysotracker positive particle trafficking in control axons. And to our surprise, we found that this is also not affected by interferon, local interferon treatment. So indeed, whatever the local interferon treatment is doing, it is specific to alpha herpes viruses. So then we were very interested, but we were puzzled at the same time, because we knew that interferons, to have an effect, they have to engage their cognate receptors on the membrane and induce a JAK-STAT pathway to activate transcription of several interferon-induced genes. And for this to happen, you need to activate signal transducer and activator of transcription, STAT molecules. And upon their dimerization and nuclear import and activation of transcription, you can have these ISGs. So we wanted to see how this works in an axons without a nucleus. So we wanted to track these STAT molecules. So here, as I already showed in the previous slides, we treat the axons with interferon. And immediately after that, starting 1 hour, we look at the STAT molecules, and also we look at phosphostat molecules. And we found that upon interferon beta treatment, we can observe an immediate increase in phosphostat molecules in axons, but we couldn't see that much in axons after interferon gamma. But when we harvest the cell bodies, we saw that There is no phosphostat in the interferon beta treated axons, but there is phosphostat in the cell bodies where we treat axons with interferon gamma. That was very interesting, and we wanted to further check what's happening in the cell bodies. And when we stained this neuronal nucleus with phosphostat antibodies, we saw that indeed, when the axons were treated with interferon beta, there is no nuclear accumulation of phosphostat. But when the axons were treated with interferon gamma, we saw that in a number of neuronal nuclei, there is phosphostat accumulation. So phosphostat is locally retained in axons upon interferon beta treatment, but it is going to the cell body and accumulating in the neuronal nucleus when it's treated with interferon gamma. So apparently there is a two-step regulation of this, and interferon beta treated axons, the responses are local. And the axons are responding autonomously. They do not require any transcription. We still do not know what phosphostat is doing there and what it's interacting with. I am still currently investigating this. But when it's interferon gamma, the responses do not stay there. They are somehow transmitting to the neuronal nucleus, and there is a more global response to that that includes transcription. So I wanted to see the effects of this local interferon effects, because it's not completely blocking viral trafficking. It's not completely blocking the infection. It's reducing. And I was interested in understanding the consequences of inefficient virus transport in axons. And in the next set of slides, I will show you that the number of viral particles infecting axons indeed determine the outcome of virus infection, whether it will go productive or quiescent in the neuronal cell bodies. So this is a very basic setup. What I tested was I infected the axons in the end compartment with different PFUs of PRV, starting with high MOIs. With 110. And in this assay, I'm using the red capsid PRV, and it is important to know that capsid protein is a late protein, so it is only expressed after the DNA replication starts. So you need viral DNA replication to see accumulation of the red capsid protein. And as I already showed you, if I infect axons and wait 10-15 minutes, I can detect nice efficient particle motility. And this is more or less the same at 100 MOI or 10 MOI. And this is reflected as a full cell body compartment infection in 24 hours. If you look at here, all the cell bodies are infected in 24 hours when I infect axons with 110 MOI. But if I go one log below and if I infect with MOI 1, now the particle trafficking is inefficient, and I don't see any red capsid accumulation in 24 hours. But if I wait long enough, now the infection spreads. So there is an efficient infection threshold between MOI 1 and 10. But I also wanted to understand what happens if I go below 0.1 and 0.01 MOI. So if I infect axons with such a low MOI, for 20 days, I do not see any red capsid accumulation. And this is what I got after 20 days of incubation. But I wanted to figure out whether these particles are lost, whether they couldn't make their way to the neuronal cell body, or maybe the infection is cleared and there is nothing left in the S compartment. To understand that, I co-infected the S compartment neurons with a green capsid virus that is UV inactivated. And this showed me that there is still red capsid PRV silenced and retained in the cell bodies, and I could bring back their signal after co-infecting them with the UV inactivated green capsid virus. And this actually showed me that these virus infections in the chambers can be silenced and reactivated. I was not expecting to have such a reactivation system by just infecting the axons with a low MOI. So, but as I already told you, this is one of the hallmarks of alpha-herpes virus infection, that it is indeed silenced in neurons and that can be reactivated. So I was happy that we could replicate what is happening in an in vivo infection, but I wanted to characterize this further in the chamber system if I want to use this as a latency model. So I wanted to see how many of these cell bodies are infected, and if they indeed express latency transcripts and signatures of latency, basically. So in this experiment, I used a spread deficient PRV. This virus also expresses GFP, so I can count how many cell bodies are infected upon such a low PFU infection. And then you can make the proportions. For example, here you can see only the 50% of the neurons in the S compartment are connected, and only less than 1% of these neuronal cell bodies are infected and expressing GFP in this scenario. And on average, in 25 different chambers, I counted 9.8 cell bodies in the S compartment that are expressing GFP. So this number is very low, but very interestingly, it matches what is published in animal models of HSV1. Only a very small number of neurons get the quiescent genomes. And when I checked these cell bodies that are expressing GFP, and I asked the question, what is their transcription signature? And indeed, they express the latency-associated transcript much more than the lytic protein EPO transcript when compared to the productive infection. And this is indeed showing that these virus particles, when they enter the axons at a low MOI, they can establish a quiescent infection similar to latency by themselves. And this is kind of the first model system that could allow us to study not only the reactivation, but also the earlier events in the establishment of latency decision-making step, because so far, what we had with the HSV1 reactivation latency models, we had the in vitro model where you force latency by blocking DNA replication. This is also a good model to study reactivation, because then you can ask what signals are reactivating, but you cannot study the steps before the latency is established. So then I started revisiting these hypotheses that I previously mentioned, that why does silencing occur when axons are infected with small number of virions? Is it because the nucleocapsids are separated from the tegument proteins? And yes, after axonal infection, these are separated, and nucleocapsids are carried to the neuronal nucleus with 3 or 4 inner tegument proteins, but many of the other teguments are left behind, and one of them is a very important transactivator, VP16. If maybe VP16 is retained in the axon, and if it cannot make it to the neuronal nucleus at the same time as the nucleocapsids, maybe the genomes are not transcribed and they are quickly silenced by the silencing machinery. And if maybe there is large number of virions, maybe the trafficking is so efficient and some VP16 molecules can reach there and start the productive infection. And this is mostly what we see in our chambers when we infect them with 110 MOIs. It's a very, very high multiplicity of infection. So then to understand that, I started investigating escape from silencing. And again, that is a very, simplistic design. I am infecting the axons with a very low MOI of red capsid virus. And as I said, red capsid is a indicator of a productive infection. And at the same time, I'm not waiting 2 weeks, 3 weeks so that the latency can be established. I'm not assaying the reactivation. I'm just treating the cell bodies with certain inhibitors, viruses, or virus-like particles to see whether I could change the mode of infection from silence to productive. So to understand this switch early on in the neuronal cell bodies. And of course, the first things I tried, I took advantage of the West HSV1 reactivation literature. And as you know, HSV1 has 5 immediate early genes. And to activate transcription of these genes, you need the tegument VP16 interacting with host factors. And then this can start the cascade of gene transcription. We know that some of these immediate early genes, they have cyclic AMP response element or CRE element in their sequence, and certain molecules like forskolin, by binding to the adenylate cyclase on the cell membrane, they can induce conversion of ATP to cyclic AMP. And this in turn activates protein kinase A, which translocates into the nucleus to activate promoters that have these motifs. So these were the first things I tested on my system. The other one is a stress pathway, C-Jun N-terminal kinase, was also reported to have an effect on HSV reactivation. And luckily, we also had inhibitors to those. And to make the long story short, I will tell you that when I used forskolin or a cell-permeable version of cyclic AMP, I was able to activate PKA in these neurons, and I was able to induce escape from silencing for the incoming PRV particles. And this pathway indeed required active PKA and JNK kinase in the cell to induce escape. And this required 6 to 7 days for the red capsid PRV to spread in the S compartment. Then my question was, what happens if I provide viral components that is missing maybe because they are left behind in the axons? And the first thing I tried was a UV inactivated green capsid virus, as I showed you before. And if I infect cell bodies with the UV inactivated green capsid, simultaneously when infecting the axons with a low MOI red capsid virus, in 3 days, the red capsid virus can escape from silencing and spread to the whole S compartment. It is very efficient and fast. And to see this effect, you have to have the cell entry mechanism, because the virus mutants that cannot enter, they do not exert this effect. So they need to enter, and they provide either lots of teguments, or maybe due to the presence of lots of defective viral genomes, the silencing machinery is somehow saturated. Either way, this pathway does not depend on active PKA or active JNK in the cell, and it is a very fast pathway. I also tested the effect of other DNA virus infections, because I want to see whether it's a generalized response to a DNA virus infection, but it's not. It is specific to UV inactivated PRV. To be able to dissect the effects of viral tegument from the excessive defective viral genomes, I also tested the light particles in the S compartment. Light particles have the membrane and the tegument proteins, but they do not have the nucleocapsids. They do not have genomes. And they were as effective as the UV inactivated virus in the complementation assay. As soon as I filter the light particle prep, I lose this activity. So whatever is providing the escaping proteins, they are in these vesicles, in the light particles. And again, similar to the UV inactivated virus, the light particles do not require any active cellular kinase to do this. So after that, I can say that using this system, we discovered two pathways. One is a stress pathway in neurons that helps incoming PRV genomes to escape silencing, but it requires active pKa and the junk pathway and the tegument. If you provide lots of tegument to the cell bodies, it induces fast escape without the need for these cellular proteins. So then the next obvious question was to be able to tell which viral components are interacting with what cellular proteins to exert such an effect. So it is still something that I'm working in the lab, and I'm trying to identify the components, both viral and host components, and I'm doing it by using two strategies. The first one is to make several tegument null PRV or HSV mutants, and of course I'm UV inactivating them before complementing the S compartments. And at the same time, while doing the null viruses, I am producing AAV vectors expressing the tegument protein that's lacking in the UV inactivated virus. So the first tegument protein we tested and we recently published was UL13. This is a viral kinase, and it has some viral and cellular targets. And if you remember, I said that light particles do not require any active cellular kinase, and I was thinking that maybe there's a viral kinase already coming with the tegument, and that would be a really nice finding. And the initial result was very interesting. We saw that the UL13 null virus was not able to complement and induce fast escape as efficiently as the wild type UV inactivated virus in the same system I showed you in the previous slide. And the GFP tagged UL13 was as efficient as the wild type virus. So that was a good indicator that maybe indeed this kinase is the viral factor that is required to induce fast escape. And we provided this kinase alone to the neuronal cell bodies by AAV vectors. But to our surprise, we saw that it did not induce escape from latency. So our conclusion and hypothesis is that probably there's another viral tegument protein that primes these neurons for productive axonal infection upon phosphorylation by UL13. So at the moment, I'm trying to finish the studies to publish the work on the VP16, because VP16, as I told you before, is one of the key regulators of immediate early protein translation, and by itself, it did not induce escape from silencing, but probably together with UL13, we will be able to see escape from silencing. So I try to explain you that I want to understand virus-host interactions starting from the axons and try to find what regulates the mode of infection and how it is controlled in the neuronal nuclei. So far, many of my experiments, I used pseudorabies virus, but I am trying to establish the same systems with the human virus HSV1. And at the same time, because my system allows studying different pathways and also the spread of different viruses, I want to see how axonal stress responses regulate invasion of nervous system by some of these neurotropic viruses. And I am very interested in understanding some of the basic neuronal biology that we didn't know so much about so far. For example, the silent mRNAs in axons and how are they translated upon certain stimuli. And virus infection is a very good scenario to test some of the hypotheses. And as you can see, I need students in my lab to help me with this research, and I need a postdoc position, and I have a postdoc position available. If you know of somebody who is interested in neurovirology, please tell them to email me from my UCI address. And with that, I would like to thank you for listening, and I would like to thank my wonderful postdoc advisor, Lynn Enquist. He is indeed the master of pig virology, and recently he gave me my certificate of pig virology before I leave New Jersey, so I have one. And I was very lucky to work with and collaborate with many wonderful students and scientists, Jan and Margaret. They helped me a lot. Esteban, Ian, Kathleen, Yolene, Oliver, we all collaborated on many projects. And of course, I have to thank to my mass spec collaborator, Ileana, Christia, and Todd from Princeton, and David Perlman. And with that, I can take your questions and we can discuss more if you want.
That's great. Thank you so much, Orkide, for that. Great job, really. Tour de force. And now I know more about neurons than I never really thought they were this complex. So. And we're super intimate. So I think what we should do is, if you want to stop sharing your slides, Alec, you can go ahead and ask your question. Alexis, I am just going to let you just unmute and ask your question. I don't. Can
you see me?
Yeah, there you go. And Jiaming, I will get to you next.
Yeah. So yeah, I might have missed it, but can you reactivate your low MOI viruses by infecting on the axon side of your system?
Yes, there's a very good question. I didn't mention that part, but I'm also working on complementing the axons. But it is a little more complicated than just complementing, because I didn't show you the study that there is competition for injured molecules. There is also competition for trafficking. So if you add virus particles, too much virus particles in the end compartment, then they compete. And if those particles are UV inactivated, of course, they make things a little more difficult for the replication competent low MOI red capsid particles. So I cannot tell you a definitive answer about UV inactivated virus complementation, but my idea is that if there is an injury in the axon around the time maybe or after the low MOI infection happened, probably that would lead to a signaling in the cell body that could reactivate or induce escape from silencing.
Yep.
Jiangming.
Hi. Okay.
Hi.
Good to see you.
Very good to see you too. Yeah. Thank you.
So I just want to follow up your one comment. So you discussed the differences between the alpha herpes and rabies entry into the neuron cell. Can you remind me again? I think I kind of remind me what's the difference in terms of their get into the neuron.
Okay, so rabies virus particles, like many of the other neurotrophic viruses, they are endocytosed. So they are carried in an endosome. So dynein is interacting with this rabies virus endosomes. Alpha herpes virus, on the other way, they fuse their membrane with the axonal membrane. So the nucleocapsids are released. They are by themselves in the axonal cytoplasm. So they have to engage dynein motors themselves.
Okay, so that's the main difference.
Yeah.
So that's why also the quick, you mentioned after 15 minutes, you always see the red particles.
Yes, with rabies virus, we had to wait 2 hours to see any detectable particle motility. So we were very surprised to see that. We didn't expect it to take that long, but with rabies virus, you have to sit in front of the computer and wait to see the first one, and it took like 2 hours.
Thank you.
Welcome.
So Orkide, I'll ask you a question
Mhm.
while other questions trickle in. So what kind of pattern recognition receptors are expressed in neurons? I mean, how are they sensing these viruses to make the cytokines that you were able to detect?
Okay, so well, they express the nectins family. So this is how the virus infection happens, and they have the receptors for other cytokines. And we also tested the antibodies, for example, the interferon receptor. And when you treat them with the antibody against the receptor, they do not respond like that. So indeed, they have all the receptors for the interferon. One thing I do not know is the interferon lambda receptor. And this is what I'm going to test as soon as I have my system up and running. Because
do they make any of those cytokines themselves, or is
very little, because the threshold is very high in neurons. They do not respond to interferon very quickly and in a very high level. They try to keep it low level. They try to save themselves. They are pro-life. They don't want to go to apoptosis immediately when there's a virus infection. They try to fight with it with different means.
Okay. Andrea, can I get to you after Yongshen? Okay, I'm trying to monitor. So Yongshen, then Andrea, then Zainab.
Yeah. Hey, Orkide. So
Hi.
my question is, can you use your system to study why only neurons, what the virus is only silenced in neurons?
Yes, that's a good question. So it is related to indeed the neuronal machinery, because neurons are not very active. They are at G0. They do not, divide. So they stop some of the pathways, and herpes viruses, they adapt. So probably they are intrinsically wanting to be silenced in those neurons. They explored the neuronal machinery so that they can live forever in the neuron. And there were some studies before, earlier times, when neuronal culturing was not that efficient. They tried some primary fibroblasts. And if you infect with low MOI and treat them with interferon or DNA replication inhibitors, you can also achieve a state similar to neuronal latency.
Oh, I see.
So it depends what the cellular stage is. And neurons are already, I'm using primary neurons. They do not divide. They're at G0. So they are intrinsically available for such silent infections. But you can also somehow train the primary fibroblasts to act like them.
So in that case, then, because in the neuron case, I thought the part of the reason why the silencing is established is because you have this like VP16 is lagging behind,
yes,
or other tegument proteins are lagging behind.
Mm-hmm.
But if you can establish latency in, let's say, fibroblast, right?
Mm-hmm.
Then you wouldn't see this type of lagging because there's
different.
no polarity. Yeah.
So this is what I'm saying. These are actually the existing models. So they start productive infection, because whatever you do, how low MOI you can go, they start productive infection, but they block the DNA replication. So it is artificial indeed, as you say.
Mm-hmm.
But the state at the end, even if there's VP16 or not, if you block DNA replication, then they are prone to be silenced.
Okay.
So they cannot progress further. So this is why I say it's an induced latency or forced latency. Still, you can study reactivation, but the initial events that is leading to the silenced form is different.
Okay. All right. Thanks.
Mm-hmm. Yep, sure.
Andrea, the floor is yours.
So this is a little bit different. Really nice talk. Thank you very much. I was wondering, perhaps, if other people have looked at the interaction between the Alzheimer's amyloid plaque proteins and this virus, because that is also being trafficked the same way. And if there's competition and there's this big theory about perhaps interactions between the two.
Yes. That is a very good question. And for a small amount of time in my research, I tried to see if there is some transport pathway that is used similarly, or maybe alpha herpes virus particles help those to traffic. That was not easy to study because I'm not using the central nervous system neurons, and there are indeed key differences between them. So my research on that was not that successful, but in future, when I start using central nervous system neurons to see how herpes virus infection happens, it's very different. CNS infection of HSV and PNS infection proceeds differently because these neurons are intrinsically different and their innate immunity genes work differently. So maybe it would be more interesting to try to see the interaction or how one or the other affecting the other in the CNS neurons. But I'm not sure if indeed HSV1 reactivations have a actual effect on the Alzheimer's progression. Maybe it's just coincidental because they happen to be at the same areas and using the same pathways. Still looking at the literature, there is a mechanistic link missing, and it is indeed very hard to study.
Thank you.
Welcome.
Zeynep?
Hi, thank you, Orkide, for an amazing talk. This was really exciting.
Thank you.
My question was about the neuronal injury and the uncontrolled calcium influx and calmodulin activation. Have you ever looked and, saw anything that might affect this kind of triggering in that sense?
Yes, we are currently trying to understand the molecular machinery behind this local protein synthesis. And we know certain things. We know that upon entry, there is small calcium spikes in axons. It is not huge, but we can really detect it by using a calcium sensor like GChem6. And we know that protein translation starts upon calcium signaling, but we are trying to understand the molecular players in between these two. Indeed, when we infect axons, if my hypothesis is correct, it induces an injury signaling, which would in turn lead to the synthesis of retrograde injury signaling molecules.
Right, and NMDA receptor involvement and all that.
Exactly. So currently I'm trying to understand how that works, and indeed maybe the high MOI infection just resembles an injury, and this would be very clever for the virus to just use that pathway for itself. I'm trying to understand the molecular mechanism behind that for some time now. There is still one student working on it. Hopefully soon we'll be able to tell you more.
Good luck.
Thank you.
Thank you.
And Robert, do you want to ask your question or do you want me to ask it?
Ask it for me.
Okay. Robert would like to know if reactivation of the closely related virus, such VZV, so herpes zoster, could be actually caused by signals from unrelated alpha herpes viruses and injury in the same neurons. I guess I captured the essence of the question correctly.
Thank you.
That's a very good question, and this is something what I'm also very interested in. And if you ask me in a laboratory model, that should an injury signal or a different virus infection, because in my model, in the simplified in vitro model, when I infect the S compartment with an UV inactivated virus, I do see. What I didn't try was infecting them with the HSV to see whether HSV would reactivate PRV in this system. But these are all in vitro, and maybe we could test them. In vivo is a little different because where the latency is established is slightly different between HSV1, HSV2, and VZV. Their ganglia might be maybe in the same ganglia in the different neurons. So how global stress responses affect reactivation is still not known. And if you look at the animal models, it's more complicated. Sometimes they see, only, let's say, 10 ganglia out of 10,000 10 neuron in a 10,000 neuron ganglia have the latent genomes, and not all of them reactivate at the same time. So that is a very curious scenario. Is it because of these animal models? Is it like that in humans? It is not well known yet. I'm very curious, but I believe we don't have really good model systems to study these things in a controlled manner. And hopefully, maybe, we can have one sooner than later.
Well, we're at the top of the hour. We're a little behind, but I see no other questions. But I just was wondering if whoever wants to stay on, and your video. We could have our informal discussion